Right, and we'll email a link to all the registrants after the talk with this recording too.
Great. So I'll move on to introducing our presenter, Matt Volpe. So Matt is a fourth-year graduate student in the Balskis lab in the Department of Chemistry and Chemical Biology at Harvard, where he does research on focused on the chemistry performed by bacteria that live in the human gut. Originally from Philadelphia, Pennsylvania, Matt traces his interest in chemistry and biology back to his parents, who I believe are on this call, whose careers in medicine made him curious about how diseases work at the molecular level. Outside of the lab, he enjoys experimenting while he bakes, performing in plays with local community theater companies, and organizing events with our Graduate Students Union. And now I'll turn it over to Matt for his talk, Old Drugs, New Tricks, the Biochemistry of Antiviral Medications.
Thanks, Lisa, and thanks, Mark and Camille, for helping to organize everything, and welcome everyone to this installment of our COVID Night School series. Without further ado, I'm going to share my screen here, and we can dive right in. Okay, here we go. Just make sure I have my presentation working. All right, cool. So as the title implies, and as Lisa said, what I wanted to talk about today was a little bit of biochemistry about how coronavirus and some other viruses work, and how we can understand the way that some of the drugs that you might have heard about in the news might be working to treat coronavirus and stop its replication. So before we dive too deeply into anything, just want to outline a few things. First of all, I am not a medical doctor. Nothing in this talk is medical advice. I am not advocating any kind of treatment. If you experience any kind of symptoms of coronavirus or any illness, you should consult your physician and follow their advice. And it's still very important, even after we hopefully someday have a treatment and a vaccine, that we're still careful to limit the spread of coronavirus. Because, as Ben Franklin said, an ounce of prevention is worth a pound of cure. What we will be talking about here is more sort of biochemists' perspective on the drugs that are treating coronavirus and sort of what makes them interesting from a chemical perspective. So we'll be exploring how some existing antiviral drugs work, a few of them in particular, and how they might be used to treat SARS-CoV-2. And then we'll also take a little look at some of the clinical trials that are ongoing and what the data looks like right now for some of these potential treatments. I'm not going to talk about vaccines at all today because that's another really big topic, but I encourage everyone to tune in to Carolyn's talk next week, which will focus specifically on vaccines. I'm also not going to talk so much about drugs that target the human immune response to coronavirus, which is a big part of the treatment, but is again another really big topic. We can talk about that a little bit in the Q&A if people are curious, but most of the talk is going to focus more on drugs that actually target the virus itself. So just as a little sort of outlining perspective, there are more than 3,000 drug candidates at this point that have been reported in different patent databases worldwide to have activity against some form of coronavirus, so either SARS-CoV-2 or other related coronaviruses. And thus far, there is no evidence that any of these are actually improved potential outcomes and there's no data that supports any prophylactic therapy yet. But there are a ton of trials going on right now, over 600 the last time I checked on the FDA website for different types of COVID-19 interventions. So this is a huge and incredibly fast-moving area of research. So if there is anything I have missed, I apologize in advance. But hopefully there'll be a lot more data coming out in the next days and weeks, pointing to some hopeful effective treatments. So as we sort of look at the landscape of antiviral drugs, one of the things I'm going to come back to a couple of times is looking at the case study of HIV. And it's important, I think, to note because I think there are a lot of virologists and other biochemists who will get upset at me for comparing these two things, that HIV is a very, very different virus in terms of how we think about its genome and how it replicates and all of those sorts of things. But I think there are some important lessons we can learn from it, because I think it's arguably one of the most effective examples of the research community being able to produce small molecule drugs to treat human virus. And those sort of broad lessons are, I would summarize them this way, and we'll sort of come back to these throughout the talk. So one of the things we try to do, and this is sort of general for treating any kind of infection, that you want to focus on trying to develop drugs that block or affect the types of enzymes that humans don't normally have or don't need. So a lot of HIV drugs are things that target things like reverse transcriptase, which is a type of enzyme that humans don't normally make because it turns RNA into DNA, which is not something human cells normally do. Or they target specific integration proteins or other kinds of things that the human body wouldn't normally have around, because that helps us limit side effects. The other sort of big lesson is the value of combination therapies in treating viruses like this. So this is particularly important for HIV because HIV mutates pretty quickly. Coronavirus or SARS-CoV-2 fortunately does not seem to mutate that quickly, but still having a combination therapy helps limits the development of resistance by mutation and can help improve efficacy of drugs. And I think the sort of most relevant thing for this talk is that repurposing existing drugs to fight a new disease is often the best way to sort of get an initial starting point for a therapeutic intervention. So the example of this in the case of HIV was AZT was one of the first really big drugs to treat HIV. If you've seen the musical Rent, that is the drug that they're talking about in that musical. That was actually developed as a cancer drug a couple decades before that, and it didn't work. So it had sort of been left on a shelf, and then when the HIV pandemic arose, they started testing it and discovered that it worked. Had a lot of nasty side effects and wasn't super effective, so now we've gone through generations and generations of development, and I think there are something like over 40 different FDA-approved therapies. I might have to check on that number, but there are a ton of different drugs now that have really changed how we look at HIV as a disease. So I basically picked 3 general categories of drugs to look at today, and we'll look at basically just a couple examples of each to try to simplify things. And so we'll talk about protease inhibitors, RNA polymerase inhibitors, and then a drug that we're not quite sure of the effect, but looks like it might blockade viral entry into the cell, and what that might mean. So I picked these because for a couple reasons. First, that I think they illustrate some interesting chemistry that I wanted to talk about, and because all of these drugs are examples of some of the drugs that are currently going into some of the biggest clinical trials, so you're likely to hear about them more in the news. So just a quick definition before I go further. You heard me use the term small molecule earlier. So when I say small molecule, we're talking about the kinds of things that you normally take in a pill that you probably have in your medicine cabinet right now. This right here is acetaminophen or Tylenol, ibuprofen, kind of those sorts of things. So these are molecules with a few dozen atoms usually. They're smaller in mass. And these are the things we sort of think of as like a chemical, right? Like this sort of simple structure. There are also lots of drugs that are what we call macromolecules or large molecules, and these tend to be whole proteins generally. And so these are huge. You generally give them by some form of injection or IV infusion, and there will likely be some antibody-based therapies that end up coming on the market at some point for coronavirus, but we'll be focusing on the small molecule drugs right now. So in order to understand what each of these different drugs is doing, we have to understand what coronavirus itself is doing. So this is a very, very basic outline of the SARS-CoV-2, life cycle in the human cell. So you'll notice here there are a few different drugs noted on this graphic, and actually the different parts of the life cycle that they interfere with, and we'll come back to this a few times. I'm not going to talk about all of these today. So what we start with, we have a virion, a single viral particle, outside the cell. And you have this spike protein or S protein on the surface of the virus that binds to the ACE2 receptor on the surface of the cell. And then there are actually at least two different sort of mechanisms by which the virus actually enters the cell, but we won't dive too much into the nitty-gritty details of that. But it requires a human and another human enzyme to sort of help pull the virus in. Once it's inside, you essentially unwrap the virus, and what's inside that virion or that viral particle is an RNA genome. So this RNA is normally the template we use that we normally use in our cells for making proteins. And so that RNA gets translated into a series of polypeptides, which then actually have to be taken apart and then help replicate the viral genome many more times, repackage the virus into a bunch of new virion particles, and then those can be released from the cell. So the first group of drugs we're going to talk about are these. Actually, I should hold on a second. Before I go any further into the actual drugs themselves, I just want to pause and ask if there are any questions on the sort of general outline of the biology.
Yeah, so we have a couple of audience questions. Please feel free, everyone, to keep posting them in the chat. So the first was from Ned. It's a bit of a broader question. How about remdesivir with today's announcements? Do you know how this works? And I think you're going to go into that.
Remdesivir is one of the drugs we will talk about at length. Yes, so stay tuned.
And a question from Oscar, who I don't know if this is the same Oscar who joined us on the first talk. Can gamma rays destroy COVID-19 RNA?
I don't know if there's been specific research done on that, but gamma radiation, or it's a form of ionizing radiation, is very bad for nucleic acids in general. So I don't know, there's really no reason this particular virus's RNA would be any different. Now, granted, that doesn't mean anyone should go out exposing themselves to gamma radiation. It is still very, very bad for your DNA. But I would guess that if you had coronavirus, say, on a surface or something like that, gamma radiation would probably damage its RNA. Cool. All right, so let's dive into the protease inhibitors. So what is a protease, first of all? So the name protease, prote means it works on proteins, and ase is just a generic term that biochemists add to everything to indicate that it's an enzyme. So the SARS-CoV-2 main protease, this is an enzyme that's called either Mpro or 3CL-Pro, is a protein that cuts up other proteins. Isn't the whole point of the virus replicating to produce a whole bunch more proteins? Why are we cutting them up? Well, the Mpro or 3CL-Pro protease works in a very specific way. So the idea of this protein's function is that after we produce our initial set of polypeptides from the RNA through the translation process, these are actually a bunch of proteins that are produced all stuck together, and we need to separate them from each other in order for them to form a functional structure. And as an analogy, you can kind of think of this as like if you've ever had a board game where you need to pop out a bunch of cardboard pieces to actually take the pieces out and then play the game with them. They all come in this sort of organized little set where they're all attached to each other, but in their functional form, you need to separate them from that larger structure. So that's what the protease does, or what 3CL-Pro does, is pops out all the pieces, basically. So looking at it from a chemical perspective, what it means is that it's going to take a polypeptide, which is sort of a generic structure that you see here, and cut that polypeptide at a very specific point. So in case anyone doesn't remember from their high school or college chemistry or biochemistry classes, if you ever took any, however long ago that might have been, what we have here is what we call the peptide backbone. So proteins are made up of amino acids linked together in a linear sequence. And so each of these little blocks, sort of from this nitrogen atom up to this oxygen atom, is one amino acid. And then these little colored balls are just sort of abbreviated versions of what we call the side chain. So another piece of the molecule that hangs off that has a little more sort of interesting functionality on it that gives each amino acid its identity. So the reason I wanted to show this is to just make note of this really specific pattern that all proteins have. So you look along the backbone, you can see this repeating structure where we go a nitrogen atom, a carbon atom, another carbon atom with this oxygen on it, and then another nitrogen. So where you see this sort of motif of this carbon, oxygen, and nitrogen group, that's an amide bond or a peptide bond. And those are the very specific kinds of bonds that proteases work on. So what does the protease itself actually look like? So this is an actual structure of the M-Pro protease. So if you've never seen one of these before, this is what we call a cartoon representation of a protein crystal structure. So it's a little difficult to see if you weren't, but if you were to untangle it, essentially each of these pieces, the blue piece and the orange piece, are one long continuous line of amino acids. And this little cartoon representation is just meant to show where the peptide backbone goes when that long line of chain of amino acids is folded up into its functional form. And then these little sort of coiled bits and bits that look like arrows are essentially a shorthand that biochemists use to communicate information about the particular way that those amino acids line up. So what I wanted to focus on here is that, so you have this much larger structure, and this is actually two different copies of M-Pro. The way it actually works is as two of them stuck together. But we have these sort of a couple amino acids in its sequence that are highlighted. These two right here that form what's called its active site. So the active site is a little pocket. So this is the same enzyme, but in a different representation. We've now zoomed in on those two residues that were highlighted. And this is what we call a surface representation. So instead of just showing that long squiggly backbone, now we're showing, if you essentially were looking at where all the different atoms stick out, what would that surface look like that it creates? And so here's where the chemistry we care about actually happens, where the amide bonds get taken apart. Just what I wanted to emphasize is that you can see how there's this sort of tight little pocket with all these different nooks and crannies around this catalytic cysteine, we call it. So what we want an inhibitor to do, at least for this type of inhibitor, is to fit into this site and block it so that the enzyme can't do its job. And that's exactly what many protease inhibitors do. So two of the most widely studied medications, at least for SARS-CoV-2 in this class, lopinavir and ritonavir, and this is ritonavir shown down here, are HIV protease inhibitors. So HIV also has a protease that has to take apart these big proteins to sort of get their functional pieces out. But it's important to note that it's a very different kind of protease. It works by a slightly different, it has a different structure and a different mechanism. But we have reason to believe, based on some early work, that some other protease inhibitors might still work to inhibit it. And the reason I wanted to talk about the peptide backbone structure specifically is that if you look closely at ritonavir, you can recognize some of the same patterns that we saw in the peptide backbone, right? So we have the same nitrogen, carbon, carbon, nitrogen pattern repeating. But what you'll notice is that in the middle here, that pattern kind of breaks down. We don't have the same nitrogen and oxygen and carbon atoms exactly where we expect them to be. So what happens is that this looks a little bit like the normal substrate of the protease, enough that it can get in and it can fit inside those nooks and crannies, but it doesn't have the same atoms in the same places as a normal peptide. So it can't do the same chemistry. So the protease can't break it apart. Now, this is what's called a substrate mimetic inhibitor. So it's actually meant to look like what normally fits in the active site. Not all drugs work that way. Some drugs look nothing like what the substrate that normally binds an enzyme, but they just happen to fit the same pockets. So that's just one example. So in terms of the actual data on lopinavir and ritonavir so far, there was at least one clinical trial that's been completed in patients with severe COVID-19, and a combination of lopinavir and ritonavir. Lopinavir is a very closely related molecule, so this is one of those combination therapies. Did not show significant improvement in severe COVID-19. It's still being tested because there are a lot of drugs where they may not make a difference after the disease has gotten very bad, but they might still be able to help if you can catch it earlier. So just last time I looked on clinicaltrials.gov, there were at least 37 other studies registered, which are using lopinavir and ritonavir or ritonavir as one of their treatment arms, including this, the World Health Organization's Solidarity trial. This is a really big drug trial that I'll mention a few times that the World Health Organization is trying to include every country in the world on, and hopefully we'll be able to, it will probably take a few months to run, but hopefully we'll be able to give us some good global information with a really big sample size on what treatments actually work. And then there's a couple other protease inhibitors people are looking at, including darunavir, which has started trials in China. There's no data on its efficacy in humans against the virus yet, but it has shown some promise in vitro. So in vitro is a term that means we have human cells growing in like a petri dish, and then we can add coronavirus to it and check whether the virus is able to replicate. And so in this case, they've shown that with these drugs, they can prevent the virus from replicating in a petri dish, but sometimes the conditions in the petri dish and in the human body can be very different. So that's our section on protease inhibitors. I'm gonna pause here again for any questions folks have about the exciting world of protease inhibitors. Do you have any questions in the chat? I can't see the chat at the moment. So.
We have a couple of questions that are a little bit more general. We can
okay, sure, we can do general ones.
Or a little later. So one, actually this one might have some spoilers, but the question is, are some classes of these drugs that you're discussing more difficult to develop than others? And if so, why? And I know you haven't finished discussing all of the classes.
Are they more difficult to develop? That's a very hard question to answer because there are different kinds of challenges you can get with different drugs. So for example, one of the big challenges in developing drugs for HIV is its rapid mutation rate. So it evolves resistance to things. That may not be a problem with coronavirus as much, but coronavirus or SARS-CoV-2 happens to, elicit this really crazy immune response that creates a different problem. And, some drugs may work very well at killing the virus, but they also have these really toxic side effects. Other drugs don't have the toxic side effects, but they don't work as well. So there are all kinds of different challenges. And I don't think that's restricted to any one class of drug. But we can talk more about that at the end. We talked about some of the other groups.
Yeah, I think this one, we have one now from Lauren in the chat that's a little more specific. You had mentioned that the enzyme that these drugs are targeting in HIV is different or has a different pocket than COVID-19. Is this why these drugs aren't working? They just aren't blocking the enzyme?
Probably. So yes, so you noticed I mentioned that catalytic cysteine residue. So this is a class of enzyme called a cysteine protease. The protease in HIV is called an aspartyl protease. I won't go too much into the difference, but basically they have slightly different chemistry in how they do that disassembly of the peptide backbone, which is important, and they have slightly different shaped pockets. So this is kind of like the classic like fitting a square peg in a round hole kind of thing, but it's not quite that bad. It's like fitting a square peg into a slightly like rhombus shaped hole. It's like close but not quite, but if it's good enough it might still be able to help patients to some extent. And that's why I mentioned we often are just looking for a starting point right now, and there will probably be several generations of drugs that are better designed to fit specifically SARS-CoV-2.
Great, thanks Matt.
Cool. All right, so let's dive into the RNA polymerase inhibitors, which we had someone hinting at a little bit earlier. Very different kind of enzyme here, but I think this is one of my favorite classes of drugs. So SARS-CoV-2 replication relies on a very unique enzyme called an RNA-dependent RNA polymerase, or RDRP, which, like I mentioned earlier, this is one of those types of enzymes that our cells don't make. Little quick review for folks who have not been in a biology class in a while on what our cells normally do. So usually we have our DNA inside our cells. These are our instructions that tell our cells how to make everything they need to survive. Normally, this gets duplicated every time a cells divide. It's how we reproduce. It's sort of our basic instructions. When we need to actually make something out of it, we have enzymes that make a copy of the DNA in a slightly different form as RNA. So from deoxyribonucleic acid to ribonucleic acid. And then we have ribosomes that take RNA and use those instructions to make proteins. This is what we call the central dogma of biology. It's not quite so central, as we'll see, because there are a bunch of annoying, weird viruses that do different things with it. So in comparison to HIV and SARS-CoV-2, retroviruses like HIV carry around an RNA copy of their genome and then use a type of enzyme that I mentioned earlier, the reverse transcriptase, to take RNA and make a DNA copy of it, and then take that DNA and copy it into the human genome, so that then the normal human processes, represented by the blue arrows, take over and make more copies of the HIV virus. Now, SARS-CoV-2 and many other coronaviruses and other viruses in general use this RDRP enzyme to instead just make copies of RNA based on the RNA template, and then again let the human processes and ribosomes take over the process of turning those RNA instructions into proteins. Since this is a type of enzyme that humans don't have, we don't have anything that's designed to take RNA and build more RNA on top of it. It means that it might have some unique features or a unique pocket that we can try to target that hopefully won't negatively impact any other human enzymes in the process. So this is what's going on down here. As someone alluded to earlier, remdesivir is one of these ones that we're going to talk about. And this is where what really amplifies the copy number of those viruses. So there are a few different drugs in this class that all look a little bit different. A couple of the ones are shown down here, including remdesivir, ribavirin, and favipiravir. So all of these were developed to fight other kinds of diseases. But what makes them all very interesting to me is that these are all what's called prodrugs. So in all of these cases, what you see here on the slide is not actually the active species that's responsible for inhibiting the virus. What happens is that these get into your cells, and then your cells actually take it and transform it into something else that is then able to fight the virus. So we'll look at that in a little more detail for remdesivir. So in order to understand that, I'm going to review just a little bit of basic biology and how we build RNA. So RNA, as I mentioned, ribonucleic acid, it's a polymer, and it's built out of these building blocks called nucleotides. So this is arguably one of the most famous nucleotides, ATP. If anyone remembers from their biology classes, the mitochondria is the powerhouse of the cell because it produces all of this ATP. It's what our cells use for energy, but it's also what we use for building RNA. And it has these has a couple key features. So on the left-hand side here, we have a triphosphate group. So these are 3 phosphorus atoms, and when you see phosphorus surrounded by these oxygens, that's phosphate. And we have them all stuck together. These phosphate groups don't really like to be stuck together like this. So this is a very high energy, very reactive part of the molecule. That's what makes this a good energy carrier for the cell. It also what makes it good as a building block for RNA. Then we have the ribose, and that's the part where we get the name ribonucleic acid, which sort of forms the core structural piece of this. And then what we call the base on the right-hand side of the molecule. And this is what gives us our letter in the code, right? Our A, T, C, or G. In this case, what I've shown here is adenine, ATP, adenine triphosphate, or adenosine triphosphate, excuse me. So this is the A in the genetic code. I should also mention, I shouldn't say T. T is what we use in DNA. We actually use something different in RNA, but that's a conversation for another time. So normally, when we're making when our cells are making RNA, we take these nucleotide triphosphates, where we have these 3 phosphate groups on there, and then we have RNA polymerases that are responsible for actually linking these up in a sequence. And normally what we would have is there's a whole piece of DNA on the other side of them that the enzyme is holding on to that it's using as a template to decide, okay, should I put A, T, C, or G here? For simplicity, we're just looking at the RNA part here. And so what it does that by taking this oxygen atom that's hanging off the bottom of the ribose and linking it to this high energy triphosphate group that is on the sort of top half of the next base in the sequence. And so then we get this long polymer that is, many, many, many nucleotides long that encodes all of our information for how we make proteins. Now, what remdesivir does is that it actually interrupts this process. So remdesivir, if we take a look at it again, you'll notice that it looks a lot like ATP in some ways. We have this thing up here that looks a little bit like adenine. We have a thing that looks like ribose in the middle. We have this weird little bit hanging off the side, and then this phosphate group up here with some decoration on it. And so what happens is when it gets into your cells, there are a bunch of enzymes that will sort of take off all of this other stuff that's around here, and will then actually put the phosphate groups on it. So now you have something that actually looks like a substrate that RNA polymerases can use to build more RNA. And when it tries to do that, what happens is it actually takes this new version of remdesivir that your cells have made, and it actually puts it into the RNA. And the problem is it sort of looks okay at first, but basically because we have it looks a little different up here, and we have this extra bit sticking off, essentially the RNA polymerase gets stuck. And what makes this a good drug is that this molecule is built in such a way that this RDRP, or RNA-dependent RNA polymerase, will pick up remdesivir and use it and get stuck, but your enzymes, your RNA polymerases that are making your RNA from your DNA won't pick it up, or at least will pick it up much less, so that you aren't killing your own cells. So that's the basic outline of how remdesivir works. The other 2 drugs I mentioned are believed to work in similar ways. Favipiravir, there's some indications it might have other targets, but the same idea is that they essentially get elaborated on by your cells. So looking at the actual clinical data, things are a little bit different for each of these. So ribavirin was originally an approved treatment it's been approved for other viral infections. It's shown activity against other coronaviruses, again, in vitro in this petri dish type setting. It has a lot of side effects on red blood cells and liver cells. So that makes it not the most attractive drug candidate in some people's opinions. Favipiravir is approved in Japan for treating influenza. We don't have a lot of clinical data for treating COVID-19 with it yet, but it is a bit safer than ribavirin, and there are some trials going on with it now to try to evaluate that. And then, as someone mentioned earlier, remdesivir is one of the drugs you've probably seen a lot of in the news. It was originally designed to, or studied to fight Ebola, but actually was found to be ineffective against Ebola. Its safety profile looks pretty good by most people's standards. And of this group of drugs, when we evaluate it in vitro in that petri dish, it has the best potency for blocking the replication of SARS-CoV-2. There are a lot of clinical trials planned with remdesivir. it will also be another one of the drugs included in that World Health Organization Solidarity trial. There was, this might have been the data that someone was referring to earlier, but there was a release of some data from a trial that did not show, that claimed that there was not a benefit of remdesivir in that treatment group. The company, Gilead, that makes the drug, released a statement saying that essentially the treatment group was too small and the study was incomplete, and they're still continuing to study it, and they're optimistic. But that data was not actually reviewed, and so it hasn't really undergone thorough evaluation yet. So we're still sort of waiting to get better numbers on that. So I'll pause there and ask if there are any questions about the RNA polymerase inhibitors.
Yeah, so we have a few questions. I'm going to try to sort through the ones that are probably best answered now. So Lynn Beacon made a comment, remdesivir was FDA approved just now. If that's...
So just missed that?
I don't know.
That surprises me. I'll have to look more into that. Remember, there is a difference between approval and sort of other types of emergency use authorization. I would be shocked if there was enough data between the last time I looked and now for it to actually be approved. But they may have sort of promoted its status. So currently, or at least last time I checked, remdesivir was, because it made it through phase 1 and phase 2 trials, which are where you're just evaluating the safety of the molecule, for Ebola back in 4 or 5 years ago, the FDA has said it is okay to use for compassionate use or in clinical trials. So in cases where doctors think a patient is in serious danger and there's really no better option and the benefits outweigh the risks, doctors are allowed to ask for remdesivir and get it on this sort of special condition.
Yeah, so people are posting in the chat. There was a lot of news today with the results from that leak trial, 2 more studies without data that look promising from the US. The FDA granted an EUA.
Right, so EUA is the emergency use authorization, which is not the same as full approval, where the FDA has said, we actually approve this, we have data that shows that it actually works, but essentially saying it looks safe, it looks like it probably could work, and because this is an emergency situation, we would rather get people the drugs. I guess I was too busy looking at the slides and did not tune into the briefing today. And I suppose I should have.
Audience members are for, and thanks to Jane, who also posted a link with more information.
Great, well, thank you. As I said, this is a very fast moving area where things are changing very quickly. But that's good to hear that it looks promising.
Yeah, and then Abby had another question. Are there any features of the structure of these RDRP inhibitors that predict which enzymes they inhibit?
Sorry, can you repeat that question one more time? I was just reading, yeah.
Are there any features of the structure of these RDRP inhibitors that predict which enzymes they inhibit?
That is a good question. I cannot tell you off the top of my head precisely which features, or if any, are known to kind of indicate that. But that is a common thing, people look for, is specifically like, oh, there is a residue in the human active site that this would bump into to prevent it from interacting with the human enzyme. But in the viral enzyme, there's this space here, and that's what this sort of extra group added on to remdesivir is meant to do, is to give that selectivity. And that's a very, important topic of research that every, drug company is usually doing, is that's what's called structure-guided design. So where you take one of those structures, like with the protease I showed earlier, and actually try to model in how your drug would fit into that active site, and then try to look for differences between the enzymes that you want to hit and that you don't want to hit, and try to put bumps in the right places, basically. But I can't answer specifically for these drugs what those guiding features are.
Great. We're getting a few more questions, but I feel like it might make sense to come back to them after.
Okay, great. So we'll get back to, if anyone's questions didn't get answered, we'll have more time at the end where we can talk about more. Okay. Cool. And then, so my third and last group of drugs I wanted to talk about, because it has been covered so much in the news, are chloroquine and hydroxychloroquine. So these drugs, this is hydroxychloroquine shown down at the bottom. Chloroquine is very closely related. You just remove this oxygen atom. These are both originally anti-malarial drugs, but are now more widely used to treat chronic inflammatory conditions like rheumatoid arthritis and lupus. So it looks like basically that these drugs may hit a bunch of different things. And one of their effects, since it appears that they do have this benefit for people with chronic inflammatory conditions, is that if they lower inflammation, they may lower the immune response to the coronavirus to sort of decrease some of those symptoms that arise from a lot of inflammation in the lungs. I want to emphasize that there is not a lot of solid evidence on exactly what they hit, but that there have been a lot of suggestions of different things they could hit. And so that includes being involved, they may inhibit endosomal acidification, which is sort of this process after you bring the virus into the cell, that endosome, as it's called, the bubble that forms around the virus, needs to open up so that the virus can get out into the cell and actually start doing its thing. If that endosome stays closed, the virus won't be able to get out. There's also a process called protein glycosylation. So a lot of the proteins that the coronavirus hijacks in the cell actually have a bunch of sugars stuck all over them. And these sugars can be really important for their function because they change the shape and structure of the protein a bit. And there's been some evidence that these drugs might be able to interfere with that. And then also, as I mentioned, the immune response. Since one of the real problems with SARS-CoV-2 is that it elicits this incredibly aggressive immune response. You can almost think of it as a kind of overreaction of the immune system. So calming down the immune system looks like it might be beneficial. And so that gets into more of these sort of like anti-IL-6 drugs and things like that that I won't be talking about as much today. So there were some early reports, specifically from a study in France, that suggested there might have been some benefit from these drugs, but the trials were very small. They weren't super well controlled. There were a bunch of people that actually got taken out of the trial early because of off-target toxicity of these drugs. So as we were just talking about, this emergency use authorization, the FDA permitted an EUA for hydroxychloroquine and hydroxychloroquine on March 28th. A couple days ago, you might have seen that the FDA then issued they didn't walk back the EUA, it is still approved, but they did issue safety guidance essentially saying that you should be careful, this should not be used outside of a hospital setting for SARS-CoV-2. It's been shown to cause a number of different heart problems, specifically one called QT prolongation, which means that it basically changes the rhythm of your heartbeat. Azithromycin, which is actually an antibiotic, but looks like it might also have some anti-inflammatory and kind of other off-target effects, has also been used in some studies in combination with hydroxychloroquine. Unfortunately, it looks like it might actually compound that heart problem. So all this is not to say that there is any clear evidence. There was one trial that came out more recently that looked like it might not work. There's another study that came out from China recently that's still in the sort of pre-review stages that looks like it does work. So we sort of keep going back and forth a bit on this. There are a lot of trials ongoing, including using it as a potential prophylactic among healthcare workers. This probably has the most trials of any one drug on clinicaltrials.gov, though I didn't check that carefully, but there are a lot. So there was a paper that made a bit of a splash a few days ago because it was posted on one of these preprint servers where the data hasn't been peer reviewed or reviewed by the FDA yet, but the people who ran this study suggested that there was some benefit. And then this will also be another treatment that will be included in this WHO Global Solidarity Trial. So for the time being, the FDA has basically recommended a lot of caution in using chloroquine and hydroxychloroquine because of these off-target effects, especially for people who have existing heart problems. There's a sort of another issue tied up in this, which is that there are other people who also need the drug because it's what they take for rheumatoid arthritis or lupus, and there have been discussions about availability. So for the time being, as with the other drugs, it's still being studied. But it is an important sort of lesson to take from this, is that just because something, we may not know exactly how it works, or because it might be doing multiple things, doesn't mean it can't work as a drug. There are certainly plenty of drugs where we don't know exactly how it works. But you know, there are a lot of different groups that are trying to understand anything or everything and how it might work. That is the last group of drugs specifically that I wanted to talk about, but just to come back to kind of some of these sort of general themes we can take away from them. So, you know, understanding I hope one of the things people take away from this is that understanding these chemical processes that are at work in viral replication are really helpful in developing better therapies. And hopefully one of these drugs, or one that I haven't talked about today, will be able to be repurposed and show some benefit in coronavirus treatment, but we don't know yet. But that will probably only be a beginning. My guess would be that there will probably end up being a couple iterations, some improvements, maybe even combination therapies. We don't know yet because we just don't know what will work, or you know, how the disease will spread. So for the time being, there is nothing that is actually approved specifically to treat coronavirus. Even giving the news we got about remdesivir today, these are still just sort of provisional approvals to address the emergency circumstances. But hopefully we will have one relatively soon. And last but not least, of course, biochemistry is really cool, and I hope everyone feels the way I do. But it's okay if you don't. So with that, I think these slides will be posted or shared with everyone afterward. These are a couple of the different resources that were used in preparing this talk, along with some others that are cited elsewhere. A couple different reviews from the AMA and from the American Chemical Society. Harvard Medical School students put together this COVID-19 curriculum, which is constantly being updated and is really excellent. You can go on clinicaltrials.gov to look at what trials are currently running. And now there's even a Wikipedia page specifically for COVID-19 drug repurposing research. So it is a very active and ongoing research area. So with that, I just want to say a quick thank you to these folks here and the other HGSU organizers that helped put together this whole series. The medical students that I mentioned that have put together this COVID-19 curriculum, everyone, all the healthcare professionals who are out there either treating COVID-19 or treating other things and keeping us safe and healthy, the researchers that are still going into lab. I have not been able to go into lab for the last 6 weeks, but lots of people have because they're still working on coronavirus. And I'm very grateful to them for doing what they do. All of you for staying home and social distancing and coming to this instead of going out somewhere. You're helping to flatten the curve, so thank you. And then all the people who are still going to work and keeping the grocery store shelves full and things like that. We wouldn't be functioning without them. So with that, I'm happy to sort of open it up to questions in general. And thank you guys all for your time.
Thanks, Matt. That's great.
I'm going to unshare my screen at this point, but I can share it again if we want to show anything visual.
Yeah, great. Okay, so to go back to some of the questions we received before, Carolyn asks, how difficult is it to scale up production of small molecule drugs to meet the needs of a global pandemic?
Ooh, that is an excellent question. The answer, unfortunately, as I said earlier with another one, is that it varies a lot, but generally hard. So I was actually, as an undergrad, I majored in chemical engineering. And I actually spent a summer working at Merck trying to do just that, scaling up production of this small molecule drug for a larger drug trial. That was actually for an antiviral medication. It's a big undertaking. It is a very active, very, sort of specialized area of research, is how do you take something and, that works in a tiny little flask and make that reaction work at thousands of liters scale. There are most pharma companies have multiple groups that are dedicated to different parts of this process. Generally, the steps you go through to make the drug when you're working at small scale are actually different than the steps you use when you're working at big scale, because there are different constraints. So that's a field called process chemistry. That's a whole separate area of development, is how you change what kind of chemistry you're doing. And then there's also a lot of requirements around keeping those drugs sufficiently clean and pure to be safe to use in people. So it's a very big area of research. I'm sure Gilead is frantically trying to scale up production of remdesivir as fast as they can. I'm sure there are people working on scaling up the synthesis of chloroquine and hydroxychloroquine and all the drugs we've talked about. But sometimes it can take years. And that's part of why this drug repurposing thing is so important, because for all the drugs I've talked about today, they have either been in use long enough or got close enough to making it to approval that these companies have already spent a lot of time figuring out those hurdles. How do you make this on really big scale? So if you're starting out with something totally new today, it's going to take a few years to actually figure out how to do all of that. Lisa, I'm going to let you sort of moderate and tell me what I should go through.
Yeah, thank you. Let's see. So Christine has a question. How do preliminary results, I'm guessing, of these trials differ by stage of disease progression? That is, which may be better for people who are less sick, moderate, severe, etc.?
So I don't think we have enough data at this point to confidently say that any one drug is better than another for a given state of disease. Unfortunately, there hasn't been enough time to run enough clinical trials yet. There's definitely more work that has been done at this point in more severe cases, because generally it's sort of, if you're a patient who, they're already on a ventilator or they're going to need to go on a ventilator soon, is sort of easier as a doctor to make the case that, we need to try something to help this patient. This patient's in a lot of danger, so the potential benefits outweigh the risks. And so there are a lot of sort of trials that have been done that way that are considered, they're observational trials. So it's not like where you enroll a bunch of people in the trial and you say, okay, these people are going to be the control group or the placebo, and this group is going to be the treatment group. A lot of that early data came from cases where doctors said, we need to do something to try to help this patient, and this looks like it might work. And so those are a little bit different. And so what we really need is some bigger randomized clinical trials that have those control groups, and those we just don't have enough data that's come out of them yet.
Great, thank you. Question from Lynn. How does zinc play into hydroxychloroquine's mechanisms? I've heard that it gets the zinc inside the cell to help kill the virus.
I am not totally sure how zinc might be involved. I have seen in other, in other drugs and treatments and things that a lot of people have suggested that zinc might help in general with your immune system, that, there are a lot of like cough drops and things that have zinc supplements in them because it's supposed to help. I don't know exactly how that would be. Zinc is used by many different enzymes. There are actually specific types of proteases called zinc proteases. So there are a lot of different things it could do. So I don't have a good answer for you on that one.
Let's see. Question from Lee. For prodrugs, is the effectiveness of the drug dependent on the individual's ability to convert it into the active form? Is there much variation in that among people?
Yeah, so that's a very good question. So I don't know how much variation there is between people. With all people, right, they're going to have different levels of different processes. It is going to depend, right? So what generally happens, one of the reasons we often make these prodrugs is that in the case of remdesivir, when we have those decorations I mentioned on the phosphate group, those often help it they keep it more stable in the sort of in its form after it's made in the factory, and then it also oftentimes makes it easier to get it across the cell membrane. And then once it's inside, generally once it's turned into that triphosphate, those are not going to be able to get back out of the cell because the cell doesn't like to release things that are, its energy carrying molecules. So what happens is you actually get this prodrug that comes in, it gets transformed, and then it gets trapped inside the cell. And what you get is an increase in what's called effective or local concentration. So my guess would be that there is going to be some variation between people, and yes, the efficacy might depend on how much of the drug you can convert. But I would also guess that at a certain point, as long as you have a certain baseline level, you can probably convert enough of it. But I haven't seen any specific studies on that.
Great. Okay, we have a question from Frank. How many virologists or epidemiologists have a biochemistry background? Do all?
I have not done a survey of virologists and epidemiologists. I would guess pretty much all virologists know a fair bit about biochemistry because they're studying the actual viruses. There are probably some epidemiologists in the audience at the moment who might be able to answer better for epidemiologists. I'm sure many of them do, but I think when you're studying epidemiology, that's more a population level look. Might not be as worried about it, but I don't want to speak for the virologists or the epidemiologists. I am probably what you would refer to as a chemical biologist. So biochemistry is very much what I do.
Great. We have a few more questions on the presentation, but it might be fun to pivot to a more personal question from Jane. Oh. Can you tell us more about what molecules you're currently studying and what you don't know about them that you're excited to find out?
So I think as I mentioned in the little intro, so I work on the human gut microbiome. So I don't work on viruses at all. I work on bacteria, actually, bacteria and people. And in my case, I study certain types of E. coli, which you might have heard of because they're around in the environment and they live in all our guts. They produce this very interesting chemical called colibactin. It's this very complex, it's called a secondary metabolite. We think it might be linked to cancer development because it damages DNA and has all these other sort of interesting effects. It's very interesting chemistry and how this thing is made. And so I study kind of why is this thing made, what impacts might it have, and what risk might people who have this bacteria in their guts be at for developing colorectal cancer.
Great. We can come back to that at the end if there's time too. Have some more questions directly related to the presentation as well.
Sure. Yeah.
Okay, so question that just came in from Catherine. I wonder if it is worth it to apply these drugs massively for mild cases.
It might be. That's, as I mentioned earlier, having patients at different disease stages when you test the drug is important because it may be more effective earlier rather than later. But it's a tricky balance. If you want to run really big clinical trials, you have to have a lot of the drug on hand. And there's that risk that if you don't have enough of it available, you won't be able to use it for sort of these emergency cases when people have really serious disease. So it's kind of a delicate balancing act because obviously you do want to know, okay, if we give this earlier, might it be more effective? But if you have lots and lots of mild cases and only a few really serious cases, you may want to focus your drug on the people who need it most. I'm sure people will be doing more trials in mild cases, but it's not generally a good idea to just start prescribing it to everybody, because then, as we saw with the chloroquine and hydroxychloroquine case, you can create shortages of the drug for the people who might need it for other conditions.
Great, thanks. Another question, when developing drugs, especially thinking about RNA polymerases that rely on remdesivir being produced again reliably by your own cells, are you worried about mutations that occur as cells divide within the body, not faithfully reproducing the remdesivir copies?
So the thing about remdesivir is that your cells don't actually need to, they're not sort of copying the remdesivir itself. You're constantly, or you would probably take remdesivir, probably several doses of it, so that you would keep infusing it into your cells, and then your body would be responsible for, as it doing those transformations, taking off that phosphate group and then putting the new phosphate groups on. Those enzymes are very sort of core to the function of your body. Making these nucleotides is like one of those really fundamental processes that your body has to be able to get right. So chances are, if you get a mutation that where you lose one of those enzymes or lose its function, that cell probably has much bigger problems to worry about. Now, there's certainly a concern about the viral genes mutating and developing resistance to that gene. Or resistance to that drug, excuse me. So as I mentioned, you have these little parts kind of sticking out in different places where you might bump into the protein. If it changes one of the amino acids at that position in the protein, and now remdesivir no longer fits into the active site, that's resistance, so it's not being affected by it anymore. And that's certainly a concern. There were at least some studies. I don't want to misquote, but there was at least one study I was reading. I forget which drug it was, but one of the antiviral drugs that they had tested against MERS, which is another coronavirus, and a mouse hepatitis virus. They found that certain viruses could evolve resistance, but that evolving resistance actually made the virus less virulent. So it actually didn't infect things effectively, so the mutation didn't hold. Now, the problem with, for example, HIV, that you often see HIV evolving resistance to something, but it's now still just as capable of infecting. So it just keeps on reproducing.
Great. We have a few more questions from Norm. So the first one is, how many RNA polymerases are known and how many proteases? What do they share in common and what are their differences?
Tons and tons of both of them. So the RNA polymerases are a little more specific group. So you have your sort of normal human or other mammalian RNA polymerases. We actually have a few of them, but all they do is just take DNA and make an RNA copy. But when you get more out into like the world of like bacteria and then into viruses, you can get all kinds of different versions of RNA polymerases. And viruses are weird. I mean, viruses, like, just as we saw, they turn RNA back into DNA, they take RNA and make more RNA copies of it. So all of these RNA polymerases can do that differently. And there are many, many different, there are many different types of coronaviruses, many different types of retroviruses. So all of those things they're all gonna have slightly different polymerases. Proteases are actually an even, well, I don't want to say this for certain because I haven't looked at the exact numbers, but I guess you could think of it as an even more diverse class, because humans themselves produce hundreds or probably thousands of proteases, maybe not thousands. But they all have different sort of things they look for and where they clip different proteins, and they're all important for different functions. So I guess that didn't really answer your question, Norm, but there are a lot. So I think the main things we worry about when we're looking at these drugs is what class of protease it falls into, for example. So as I mentioned, there's the cysteine proteases, which is what coronavirus's Mpro is, but there are also the aspartyl proteases, which is what HIV's protease is. Then you also have serine proteases, and a few others as well. So those are like the main groups we worry about. They all work a little bit differently.
Great. Second question from Norm. What structurally makes SARS-CoV-2 target ACE2 receptors?
It's basic. I can't tell you specifically like what parts of the protein are responsible. There have been a lot of structural studies on that, and it's been a big area of research. But basically, it's just a matter of what the spike protein, that S protein, fits into. These proteins have all kinds of different little bumps and ridges, and they have different negatively charged and positively charged groups sticking off. And so you have to find something else whose bumps and ridges fit into your bumps and ridges. And it just so happens that the spike protein has evolved to fit the bumps and ridges that are on ACE2. And that's the same receptor that the SARS virus that emerged several years ago, the sort of original SARS, uses, and that many other viruses use as well.
Okay, so we have a couple more questions in the pipeline. I would just encourage anyone who has a burning question to type it in the chat now in the next couple minutes. But let's see if we can get through the current questions first. So one question is drug research shared between groups in different institutions and countries, or do you typically have to wait until results are published even during unprecedented times like these?
It can vary a lot. It's going to depend on the specific individuals doing that research, how extensively they want to share it. I think right now it seems like people are sharing things pretty widely because I think they recognize that we really need to get something that will work. But things are also getting published much faster. I think a lot of journals have started their editorial board has started kind of like fast tracking things that are coronavirus related and look particularly important. So normally when you submit a paper to a sort of very high profile journal, you might submit it and it's going to be like 6 to 9 months before that paper actually comes out. You have to go through this review process and all kinds of things. But with big coronavirus papers, we've seen them coming out in like days or weeks. So it's also a matter of, the journals are sort of going through the peer review process, are trying to share that more widely. Now, if you're at a pharmaceutical company, my bet is that they are probably not sharing things super widely, just because working at a company is different and you want to protect your intellectual property, and that's sort of how the system works. But I do know that, I think everyone has sort of recognized that it is a very unique moment that we're in that requires more collaboration even than we normally see.
Yeah. As a follow-up to that, what impact do you think that would have on the quality of research and publications coming out right now?
It can be kind of a double-edged sword, I think. I can't give any specific examples at this point, but obviously, you can imagine, very pressing times, a lot of attention on this coronavirus thing might encourage, sort of push people to be kind of hasty and publish something before it's really ready. But at the same time, I think sort of having this unique opportunity where so many people from so many different fields and backgrounds and institutions are trying to work together to do this might really, help in a big way. So I think it depends on sort of scientists continuing to be sort of careful and thoughtful about what they're doing and trusting the peer review process, that sort of everyone is trying to sort of check each other carefully and making sure we're doing everything in a quick but thoughtful way. I saw a comparison at one point where someone looking at when HIV was first identified and when we first isolated the virus and actually crystallized one of its proteins so that we could get a structure of it, versus the SARS-CoV-2 virus. For HIV, it was years because research was different. We didn't have the same technologies, just a very different time. For SARS-CoV-2, we first identified it in November of last year. We'd isolated the virus days later, I think, and we had a crystal structure of some of the proteins in a couple weeks. And that was published in Science. And so that was just an unbelievable acceleration of research.
Well, I think that brings it to the end of our questions. Thanks, Matt. That was a really great presentation. So we did record this talk. We will be sending out the link to everyone who wants to rewatch or could make
I
it.
hope I didn't say anything stupid.
We will edit out anything you said, Matt, that you.
Oh, thanks.
We'll also share the slides if that's okay. Is that all right?
Yes.
Okay, great.
Make sure there are no typos.
Yeah. Okay, so while we prepare all of that, okay, Jane is also asking if we can include a link to Matt's research.
Sure.
We can definitely do that. Should have done it for our previous speakers as well. They're all doing really cool stuff. So next week, Carolyn Boudreau, our virologist, is going to be speaking about developing a vaccine for COVID-19, which I'm sure many of you will be interested in. So we hope you'll join us next Wednesday at 8 p.m., I think. Yeah, anyone else have anything else to add?
Thank you all for attending, and hope you all enjoyed some biochemistry. I certainly did.